When we contemplate teeth, we instinctively picture the formidable armaments within the mouth, tools for mastication and predation. Yet, the evolutionary narrative of these indispensable structures begins not in the oral cavity, but scattered across the external surfaces of ancient organisms. Imagine the Cambrian seas, a crucible of evolutionary innovation and escalating ecological pressures. Here, the precursors to teeth first emerged, not as feeding apparatuses, but as microscopic dentine-tipped tubercles embedded within the heavy dermal armor of stem-vertebrates and arthropods some 520 million years ago. These early odontodes were distributed across the skin, serving a primary role in sensing the surrounding environment. They were exquisitely tuned to detect subtle shifts in water flow, pressure gradients, and the proximity of potential predators, functioning as a primitive sensory array. Concurrently, eel-like vertebrates, known as conodonts, developed crown-shaped phosphatic bars deep within their pharyngeal region around 505 million years ago. These structures were likely employed for filtering and grasping planktonic prey, representing an early distinct lineage of hard-feeding parts. Later, jawless ostracoderms evolved dermal denticles and rudimentary oral plates positioned around the lip margin. These allowed for nipping at soft prey or scraping algae, marking a tentative step towards utilizing hard tissues near the mouth. The very genesis of this hard tissue, the dentine and enamel-like cap of the odontode, was a pivotal evolutionary event. Driven by the intense predator-prey dynamics of the Cambrian, several lineages of armored fish responded by reinforcing their bony integument. Small buds of odontode tissue began to protrude through this dermal armor. Intriguingly, the genetic machinery orchestrating this development involved the same fundamental odontogenic gene network, including key signaling pathways like BMP, FGF, WNT, and SHAR that still governs tooth patterning in modern vertebrates, including ourselves. This ancient genetic toolkit, activated within the dermis, initiated the formation of these hard buds. Crucially, these early odontodes were intimately wired to sensory nerves. Their dentine cores were permeated by microcanals, transmitting signals related to vibration and water pressure, effectively functioning as a rudimentary lateral line system. The true revolution in dental evolution occurred with the advent of jaws. Evolving from the anterior gill arches in late Silurian fish, the hinged jaw provided a novel functional domain, a mobile aperture capable of capturing and manipulating prey. With the emergence of jaws, the odontode-producing cells, which migrate during embryonic development via neural crest cells, were free to colonize the inner surfaces of this new structure. Wherever these migratory cells settled, they retained their inherent capacity to sprout enamel-capped spikes, the first true oral teeth. Natural selection then commenced its meticulous work, refining the shape, size, and arrangement of these nascent teeth. Different lineages adapted their dental batteries to exploit specific ecological niches, resulting in the diverse forms we observe today, from the crushing plates of placoderms to the zip-like shearing blades of sharks. The subsequent evolutionary trajectory saw vertebrates adopt myriad strategies regarding their dentition. Some lineages retained and elaborated upon teeth, while others reduced or entirely lost them, often repurposing the underlying genetic machinery. Birds, for instance, transitioned to lightweight keratinous beaks, an adaptation facilitating flight. Although their tooth-forming genes persist within their genome, they are typically silenced, a remarkable example of genetic potential lying dormant. Turtles similarly replaced teeth with hornitomia, effective for processing aquatic vegetation. Baleen whales offer a spectacular case of developmental repurposing. Their embryos initially develop tooth buds, which are subsequently reabsorbed as keratinous baleen plates form, filtering vast quantities of small prey. While many fish and some amphibians continuously replace worn teeth throughout life, mammals largely sacrifice this perpetual renewal for enhanced occlusal precision, developing only two sets of teeth, deciduous and permanent, that interlock with remarkable accuracy. The saga of tooth evolution encapsulates several profound principles applicable across diverse evolutionary problems. Firstly, it exemplifies the concept of origin outside the functional domain. Innovations often arise in one context, like sensory perception, only to be co-opted and refined for an entirely different purpose, such as feeding. Secondly, it highlights the power of modular genetic toolkits. Once an organism possesses the fundamental odontogenic gene network, the potential to develop tooth-like structures becomes highly portable, capable of being deployed wherever developmental signals permit. Finally, the story underscores how ecological arms races serve as potent drivers of hard-part elaboration. In environments characterized by intense predator-prey interactions, the selective pressures favor the rapid diversification and refinement of structures like teeth, essential for capture, defense, and processing resources. Despite the remarkable clarity gained from the fossil record and developmental biology, certain questions continue to stimulate scientific debate. Were the phosphatic structures of conodonts truly homologous to vertebrate teeth, or did they represent an independent evolutionary experiment in hard-feeding structures? Did the oral plates of ostracoderms function primarily for biting, or were they limited to scraping substrates? And within early mammals, what selective pressures favored the evolution of complex tribosphenic molars in some lineages, while others, like the multi-tuberculates, developed rodent-like chiseling incisors? For those eager to delve deeper, fascinating rabbit holes abound. Investigating genomic knockouts that can reawaken tooth development in chickens, echoing the famous Collar and Fisher chickenodont experiments, offers insights into genetic potential. Exploring how deep-sea dragonfish evolved transparent fangs with unique nanostructured enamel reveals the extremes of adaptation. Or perhaps examining the fossil record of ever-growing rodent incisors and the intricate mathematics governing their exponential growth versus wear rates. Each of these avenues offers a glimpse into the intricate, dynamic, and often surprising journey of dental evolution.